Hello, Dr. Hamid is here. Today we are going to talk about cholinergic agonists. Cholinergic agonists are the chemicals which can increase the effect of acetylcholine in the synapses. They have different mechanism of action, and today we're going to know about the mechanism of action, the different groups which can increase the effect of acetylcholine in the synapses. Before talking about these drugs, first we should know about the neurotransmission in the cholinergic fibers. We should know about the synthesis, we should know about the releasing, as well as we should know about the metabolism of acetylcholine in the synapses and cholinergic fibers. Neurotransmission in the cholinergic fibers has 6 steps. The first step for neurotransmission in the cholinergic fibers is synthesis of acetylcholine. For synthesis of acetylcholine, we need 2 substrates. The first substrate is acetyl-CoA. The source for these acetyl-CoA are mitochondria, which are located in the nerve terminals. We have lots of mitochondria in the nerve terminals, and these are the source for acetyl-CoA. The second substrate for synthesis of acetyl-CoA is choline, but we do not have choline in the nerve terminal. We have to uptake the choline from extracellular fluid or extracellular environment. So choline are in the extracellular environment. One of the characteristics of choline is that it has positive charge. Because of this positive charge, choline is not able to cross the cell membrane, and we cannot transport this choline without a transporter. The transporter for this choline is choline sodium cotransporter. Choline sodium cotransporter, which co-transport choline with sodium. So we transport choline inside the cell, inside the nerve terminals, choline, as well as we transport one sodium inside the nerve terminals. This step, uptake of choline or transport of choline from extracellular environment or extracellular fluid can be inhibited by one chemical which is hemicholine. Hemicholine, it can inhibit the first step of synthesis of acetylcholine. And this step, uptaking of choline from extracellular environment is the rate limiting steps. It means that if we can inhibit this step by using of hemicholine, we can inhibit the whole process of neurotransmission of cholinergic fibers. The second step for synthesis of acetylcholine is by the help of one enzyme which is choline acetyl transferase. Choline acetyl transferase. And these 2 substrates, we make acetylcholine. So acetylcholine is made of choline and acetyl-CoA. Now we know about the source of acetyl-CoA and choline by the help of choline acetyl transferase. These enzymes add one acetyl to the choline and make acetylcholine. The second step, this was the first step of neurotransmission in the cholinergic neuron, which is synthesis of acetylcholine. Synthesis of acetylcholine is the first step in the neurotransmission of cholinergic fibers. The second step of neurotransmission of cholinergic fibers, we have vesicles in the nerve terminal. In the cell membrane of vesicles, we have a transporter which is VAT. VAT is for vesicular associated transporter. By the help of these vesicular associated transporter, we package acetylcholine in the vesicles and we store this acetylcholine in the vesicles. Acetylcholine is the main neurotransmitter in the cholinergic fibers, and we store it in the vesicles by the help of VAT or vesicle associated transporter. Now, we have some chemicals which can inhibit or which can block this transporter, and by blocking this transporter, we can inhibit storing and packaging of acetylcholine in the vesicles. And this chemical shows anticholinergic effect. The name of this chemical is hemicholineum, vesamicol. Vesamicol can blocks the VAT. By blocking the VAT, it inhibits packaging and storing of acetylcholine in the vesicles. We have some other chemicals also in the vesicles which are co-transmitters. Co-transmitters are in the cholinergic fibers ATP as well as protoglycan. These are the co-transmitters which we store these co-transmitters with acetylcholine. This is the second step in the neurotransmission of cholinergic fibers. So the second step is storage of acetylcholine. Storage of acetylcholine. The third step of neurotransmission, these acetylcholine will remain in the vesicles till action potential reaches the nerve terminals. When action potential reaches the nerve terminals, it will open some channels which we have in the nerve terminal, and these channels are voltage sensitive calcium channel. These calcium channels are sensitive to voltage, and when action potential reaches the nerve terminal, it can open the voltage sensitive calcium channel, and by opening of this channel, we can see influx of calcium inside the nerve terminals. Then this calcium, which is inside the nerve terminals now, it will promote fusion of these vesicles to the nerve terminal membrane, and by fusion of these vesicles to the membrane, we can see releasing of the neurotransmitter and co-neurotransmitter which are in the vesicles. Now the acetylcholine is in the vesicles, is in the synapses. This is the third step of neurotransmission. The third step is releasing, and this third step we can inhibit it by some chemicals, which is botulinum toxin. This toxin, it can inhibit releasing of. First, it can inhibit fusion of these vesicles with the membrane, and therefore it can inhibit releasing of neurotransmitter in the synapses. Some other chemicals also are there which can increase fusion and which can increase the releasing of neurotransmitter. That is the toxin of black widow toxin, black widow spider toxin. This toxin can increase the fusion, can increase the releasing of neurotransmitter and co-neurotransmitter, and it can deplete the acetylcholine storage. This was the third step in neurotransmission. The fourth step of neurotransmission, now you know about the cholinergic receptors. We had a lecture for this. Cholinergic receptors are two in types. One is muscarinic, another one is nicotinic. But muscarinic receptors are also, each subtype of receptors are also in two different subtypes. One is postsynaptic and second is presynaptic receptors. This acetylcholine, it can bind to postsynaptic or it can bind to presynaptic receptors and it can show its effect. So the fourth step in neurotransmission is binding of acetylcholine to its receptor, binding to receptor. Now we have different group of drugs which can inhibit this binding, and those are cholinergic antagonists. We will have a lecture for that also. But the action of acetylcholine will terminate rapidly by the help of one enzyme, that is acetylcholine esterase. The enzyme acetylcholine esterase will cleave the acetylcholine and it will terminate the action of acetylcholine in the synapses. Acetylcholine esterase. This will cleave the acetylcholine. It will bind first to acetylcholine. By binding to acetylcholine, it will cleave acetylcholine to choline acetate, and it will terminate the action of acetylcholine. We have two subtypes of this enzyme. One is true acetylcholine esterase and the second is pseudo acetylcholine esterase. The true acetylcholine esterase are in ganglion, central nervous system, RBC, and this is specific for acetylcholine. It's specific for acetylcholine. But the pseudocholine esterase, this is in plasma, and this is non-specific for acetylcholine. This enzyme, this subtype, it can cleave the succinylcholine. It's specific for succinylcholine. Okay, this was the fifth step of neurotransmission. The fifth step is cleavage of acetylcholine. Cleavage of acetylcholine. The sixth step in the neurotransmission of cholinergic fibers, this choline, which is a substrate of cleavage, we will reuptake it by this channel again, and by reuptaking we can make new acetylcholine. We can store this acetylcholine in the cholinergic nerve terminals till any other action potential reaches to this nerve terminal. We will release it and this cycle will occur again and again. So we will uptake this choline by this co-transporter and we will make new acetylcholine. This is the sixth step of neurotransmission of cholinergic fiber. So the sixth step is reuptake of choline. This was about neurotransmission in the cholinergic fibers.